Evidence from numerous studies points to biological formation resulting in autism disorder. Biological formation most prevalent in WNT signaling. So, what is WNT signaling? WNT signaling is a type of synaptic cell communication in the brain which features frizzled and tangled G protein coupled receptors and many different types of WNT ligands. WNT signaling is one of the factors often associated with neurological brain development and neurodevelopmental disorders such as autism. WNT signaling features a variety of WNT ligands such as WNT1, 2, 3, and 9b from the forebrain neurons in hippocampal, cortical, and striatal regions. In a normal signal transduction pathway, WNT glycoproteins bond to the extracellular space of a frizzled receptor, which activates the pathway to inhibit beta-catein phosphorylation. Before WNT proteins are bonded, a complex of molecules such as beta-catein, APC, beta-TRCP, GSK3, CKI, axin, and DVL, also known as the destruction complex, phosphorylates beta-catein, which ultimately decreases beta-catein levels in the cell. When WNT ligands bond to the frizzled receptor, it phosphorylates LRP, which connects to this destruction complex between the frizzled receptor and LRP. When DVL binds to LRP, it inhibits the destruction complex from activating, preventing beta-catein from being phosphorylated, and increases beta-catein levels in the cell. TCF genes mediate WNT signaling pathways, but when the pathway is inactive, TCF is bonded to GRUCHO, which inhibits TCF from binding to the DNA in the cell. In a normal response, when the pathway is active, the beta-catein translocates into the nucleus and removes GRUCHO from TCF and takes its place. This inscribes WNT target genes, which cause the growth and development to take place. The normal response of the signal transduction pathway causes brain development and can impact the formation of every organ system in the body. However, in an autistic person's WNT pathway, rare variants of the typical WNT2, 3, and WNT9b have been found overactivates WNT signaling. Without the standard regulation, the cytoplasmic beta-catein is degraded by the beta-catein destruction complex. Variants that decrease WNT activation can conversely lead to overaccumulation of beta-catein, which leads to the reduced sociability and repetitive behaviors linked to autism.